We normally have our meetings the fourth Tuesday of each month, but for November, we will move it up 1 week. We will discuss it as a board, but pay attention to the newsletter and check out our website also. We have some new beekeepers that are considering getting bees in the springtime. COVID kept you from doing anything. So if you are interested, what we will, we're going to start planning for next year. We normally order bees to come in April, and we will start to do meetings in January to teach people how to do beekeeping, how to keep the hive, what to look for. So if you are thinking about doing it, one of the things that we do ask people to do is you have to be a member to purchase from our club. But if you are interested, keep up with the newsletter because the information will be in the newsletter about how to purchase bees for the springtime. And those of you who just need to replace a queen, we will also have information about queens in the springtime. So with no further ado, I'm going to have. Oh, I got an announcement. Oh,
go ahead.
Well, no, go ahead.
There's 2 free bee suits back there. They are either large or extra large. Which was extra?
I think they're extra large.
Extra large. Okay, we had one of our members pass away. I think they look like they're ventilated, and in Texas, you definitely want that.
Thank you.
Thank you, Shannon. Well, it's my pleasure to our speakers tonight. She's in the last year of her doctorate. Alex Payne is her name. And I asked her to make sure she used lots of Latin terms in tonight's speech so that we didn't understand anything. And I think you simplified it a little for us. But she's come all the way from College Station, and I hope she gets home before it rains. So thanks for being here, Alex. And she'll give you lots of information if you stay afterwards and ask her.
Yeah. All right. So I think I have 2 mic situations going on right now. Can you guys hear me with that one? Yeah. Okay, so I might just be doing the one.
So it's just
yeah. I think we were having issues with the Zoom people maybe hearing.
Okay.
They're good? Okay, perfect. All right, so hello everyone. Thank you for having me. I'm excited to be back here. I think I've spoken for this bee group at least one time in the past. But I am in my 5th year at Texas A&M studying with Dr. Juliana Rangel. Some of you may have worked with our lab in the past. We like to work a lot with the beekeepers here in Texas. We just did a survey of beekeepers on how COVID impacted them. So I don't know if any of y'all participated, but if you did, thank you guys for continuing to help our lab and help the research to better know more about honeybees. So without further ado, I'm going to go into what my specialty is. Oops, sorry. Which is honeybee pathogens, and the talk can be kind of broken up into 2 parts. One, I'm just going to be going over basic biology of the pathogens and how they interact with honeybee hosts, kind of give you background on some of the more prevalent pathogens you're going to encounter while beekeeping, and then go into some best beekeeping practices in order to address your hives whenever they are infected. So, a multitude of factors that affect honeybee health. The primary ones are parasites, pathogens, pests, pesticides, climate, the environment, and nutrition. And for the sake of this talk, we're just going to focus on this big grouping right here. So honeybee pathogens can kind of be broken up into 3 major groups: fungal pathogens, bacterial, and viral. Just like any organism that has disease or pathogens that impact them, they can kind of be broken up into these 3 main groups. And then, of course, the various stressors that can cause disease outbreak to be worse, such as poor nutrition or having too many colonies in one area, as an example. So the main fungal pathogens that you'll encounter in your hives, one of them is Nosema. And before I get too far into the talk, if anyone has questions, I don't want you guys to just sit there and kind of ponder like, well, what is she saying? I don't get what she's saying. Please feel free. We're going to have a question and answer time at the end, but if something doesn't make sense or you want clarification, please just call out, and I'd love to expand on something. It's easy for me to just kind of go through, but I know everyone here is at different levels, so I would love to answer any questions along the way. So going into the fungal pathogens, first one is Nosema. There's 2 different species, Nosema apis and Nosema cerani. Here in the United States, you're going to be mainly, almost exclusively finding Nosema cerani. And what is it? It is a gut pathogen. So bees will ingest it in their food or whenever they're doing trophallaxis and interacting with other bees. They'll ingest it, and then that fungus will proliferate in their gut, and it'll cause different disease symptoms. So it typically infects adult workers. The spores come out in the feces of the bee, and then when the bees are cleaning up, they ingest it that way. So it kind of turns into bee dysentery almost. Another one is chalk brood. We call these little brood mummies because it looks like after the fungus has encapsulated them, they're these little white, dusty chalk mummies. It's typically the fungus is found in contaminated food. The spores spread that way. It infects the gut of the larvae, similar to nosema. And it does actually start out the bees, so if it gets into the colony and gets spread pretty perfectly, then it can start them out. And also it can be a carrier of viral pathogens. Fungi are large enough that they can kind of have viruses hitch a ride. You can have dual infections of different pathogens that way. The last one is stone brood. I won't say the Latin name because Chris told me not to, so sorry guys. But it's commonly found in the soil. It infects the gut of the larvae. They are pathogenic to other animals, and why it gets its name is the larvae will turn into almost like little pebbles inside of the cell. They like harden up, especially with that exoskeleton they have. They'll turn into little rocks almost. So moving on to the bacterial pathogens, how many of you guys have heard of American foul brood? All right, yeah, so I was not beekeeping at the time when foul brood was really an issue a couple of decades ago. Now it's not so much a huge concern that we're dealing with on a daily basis here in the US, but for a while it was the pathogen that was causing the big scare in the beekeeping community. It is a rod-shaped bacterium. It does form spores. It's found in the brood food, develops in the gut. You're finding a pattern, right? What makes this one so bad is that it is very efficient at surviving inside of colonies and in the environment in general. It can persist for decades on beekeeping equipment, even if it's here in Texas heat. It's a very persistent bacterium. And even if you try to use antibiotics on it, it will only knock out one state of the bacterium and it will come back eventually. So it's very, very resistant to not only antibiotics, but environmental desiccation as well. So I'll talk about how to deal with it in a second. European foul brood is its nicer sibling, but it's similar in that it's rod-shaped, but it doesn't form spores. So that's what a big difference of this. American foul brood forms spores, and you can have one bacterium that suddenly can infect thousands of cells around it. Whereas European foul brood doesn't spread as prolifically as American. It also infects and replicates in the midgut of workers, or not just workers, of adults, but again, not as bad as American. For this one, you can actually treat pretty effectively with antibiotics. However, European foul brood is one of those pathogens that a healthy colony or a strong, high population colony can pretty much deal with the infection on its own. But if you have a pretty bad infection, you can contact a vet and use oxytetracycline, as an antibiotic, and you can treat your colony that way. I actually just gave a talk to Future Vets of America, I think that was their group, and I was telling them how they might have beekeepers at some point say, hey, I need to treat for my bees, which is a thing that they didn't know about. So going over a little more, I know that's super hard to read. I don't expect you guys to. I just kind of wanted to throw this up here because of the great resources you can find online if you're having issues diagnosing a colony. The Texas Apiary Inspection Services, they have a lot of online resources and lots of pictures that can kind of give you an idea of, do I have foul brood? Do I have chalk brood, stone brood? They'll go into it, not only list the symptoms that are typically associated with that pathogen, but they'll also kind of tell you, okay, what do you need to do now depending on what it's infected with. For instance, with European foul brood, you are going to see, well, how much of the colony is infected, how strong is the colony, do I need antibiotics, like I said. Whereas American Foulbrood, their solution is we're coming out there, we might have to burn stuff. So I think that was the popular way to deal with American Foulbrood is take all of your hive equipment and destroy it to make sure it doesn't spread to not only your other colonies, but to nearby apiaries as well. So definitely use them as an example for what to check for. They have also a really cool video of the ropiness test. I don't know if you guys have heard of that, but if you're checking for American Foulbrood, one of the big symptoms is that the larvae itself melts. It's really tragic, but they give off this sulfur smell and they turn into this rubbery, like orange material as the bacterium is basically degrading them from the inside out. So you can take a toothpick, a stick, anything lying around, you put it in the cell of a larvae that you think is infected, stir it around, it will literally turn into goo, and when you pull it out, it's almost a booger texture. That's what I've been told, not my words, but it's this orangish booger. That's how you know you're dealing with American Foulbrood. European will not give you that. All right, I am partial to viral pathogens, which is not a popular thing I know to say here in COVID times, but I do work primarily with honeybee viruses. There are over 30 honeybee viruses that have been sequenced and found in honeybees as of 2020, and that number continues to rise as we have more people doing research on them and looking at more samples around the world. Viruses are the most successful pathogen group to honeybees, meaning that they are the most proficient at killing honeybees. So we have a ton of different viruses and they're constantly evolving and we are constantly having to deal with them. The 6 most common ones are these listed right here. I'm sure you guys have heard at least one of them. One of the most, I guess, iconic or most well-known is deformed wing virus. So it's those bees that you see sometimes crawling around your colony and it looks like their wings are shriveled up. And obviously they're not going to be able to fly with those wings. They emerge as adults with their wings like that, and actually within a few days, 1 or 2, their sisters will kick them out of the hive because they're deemed not able to contribute. So it can be a pretty devastating pathogen. Even bees that are infected with the pathogen but don't have the deformed wings, they can have their memory impacted, so they're less efficient foragers. They actually live a shorter period of time. So there's other less obvious symptoms that are associated with that virus. Other viruses, including black queen cell virus, if you guys do queen rearing, you might come across this, associated a lot with varroa mites, but literally the queen larvae would turn this very, very dark black color in the cell and it causes mortality, obviously. And then the different paralyses viruses. There's quite a few, and what those look like, sometimes, for instance, with Israeli acute paralysis virus, you have bees that lose all of their hair. They look like almost a completely different insect. They almost look like they've been dipped in oil. And they no longer are able to fly efficiently. In fact, a common symptom is to have a bunch of bees that are on the ground at your entrance in the grass, and it looks like they can't fly upwards. That's because it is a paralysis virus. They're not able to use their wing muscles in order to fly back into the colony. Colony collapse disorder. You guys have heard of that term. One of the big things that they think, it's still not completely known, but one of the big drivers they thought of colony collapse disorder was Israeli acute paralysis virus. Bees would go out into these agricultural operations infected with this virus, and that plus neonicotinoids made it so they could never return to the hive because they would seize up and then die in the fields that they're foraging from. So yeah, viruses plus pesticides plus environmental factors, those always come up with new scenarios we have to deal with. All right, so here are those common viruses I was talking about. You see a trend, right? Ways that they can be transmitted. Varroa are just the dirty needle of the honeybee world. I think we all know that. There are mosquito equivalent. They seem to vector just about everything. Enemy number 1, Varroa destructor, especially in the case of viral pathogens. And again, viral pathogens are one of the most devastating things to our beekeeping. So here is a graph that's showing the number of managed colonies in the US, reported in millions throughout the decades. So you can kind of see that first drop is just because they were changing their survey tactics, and that's what caused that different number. But whenever Varroa was introduced in the 80s, you can see that steady decline. And oh, you guys can see my cursor, awesome. You can see that steady decline as Varroa kind of decimated the population of honeybee colonies in the 90s. And then we have that colony collapse disorder event that happened in the early 2000s that caused that big drop off in over a 5-year period. And kind of bounced back now that we have new management techniques. So we can't talk about honeybee pathogens without talking about Varroa. I'm sorry if you guys have heard way too much about Varroa. Maybe you know all this already, but bear with me. So they feed on the, sorry, I think can you guys see the screen, like the bottom part, okay?
Yeah.
Okay. All right, so they feed on the fat body of a bee. That wasn't known until while I was a graduate student a few years ago. Before that, it was thought that it was feeding on the blood or the hemolymph. No, they're feeding on the fat body, which is responsible for a lot of hormone production of the bees. It's very important for maintaining nutritional health, for maintaining their lifespan. And you would think that's something we would know about sooner than this, right? With how long we've been working with bees and how long we've known about Varroa. But Varroa are very tricky to study. They're very difficult organisms to work with in the lab and to sequence and to understand, which is why it's been so difficult to deal with them. They do transmit viruses, like I said, while feeding. So they'll feed on one bee, go to another, and they'll take with them whatever viruses that previous bee had, and then put it in the new one, and that repeats the cycle as they continue to jump hosts until they find a cell to start laying eggs in. And here's just a quick schematic of the life cycle of the Varroa with the honeybee. So it enters into the cell with the larvae. The foundress or the mother mite will lay her eggs. She will mate with her son and then give birth to her female offspring. The son dies in there, and then the females and the mom emerge with the new adult honeybee as it's developing, and then will transmit viruses and jump to different workers. So that time period is very important for when it's in the cell with the larvae. That's when a lot of these viruses are going to be really ramped up in number, and that's when you're going to have, for instance, bees emerging with deformed wings, is when they're infected as young, immature larvae. I want to also bring up that Apis cerani is the Asian honeybee, so the Eastern honeybee. We work with the Western honeybee, right, the European honeybee. And it's similar to Apis mellifera. It's a bit smaller, much smaller population sizes overall, not really great to rear if you want a lot of honey, but they are more resistant to Varroa. In fact, they are so much more resistant to Varroa. Varroa is not an issue for this species of honeybee. The reason being is that the Asian honeybee originated in the same area and evolved with Varroa. So this area right here of Eastern Asia is where Varroa destructor originated. And it was on originally as a host, Apis cerani. Apis cerani has learned to deal with it. Suddenly, the Eastern Asian people decided they wanted to introduce the Western honeybee into Asia in order to help with their pollination services, as the Western honeybee, again, like I said, has larger population sizes and could pollinate more crops than the Eastern. So they brought in the Western honeybee, and suddenly we had the mites jumping hosts from the Eastern honeybee to the Western. And then those colonies started to swarm, spread out, and the Varroa spread out with them until eventually Varroa destructor covered wherever there were honeybees. So all over the world, minus obviously Antarctica and Australia and the Sahara Desert. So now we have, and some Hawaiian Islands. So Varroa destructor has been very efficient, very successful at switching over to this new host. And it's a host that has not had the time to evolve the same defenses that the Asian honeybee has evolved. So there it was in Eastern Asia in the 1900s. We introduced Apis mellifera in 1963 into that area. And as the bees started to spread out around the world, and we started bringing bees into South America and then North America more so, we see in the early to mid 70s that Varroa was discovered in the US. And it was confirmed in 1987 in Wisconsin and Florida. That's when we first saw Varroa destructor here in the US. So switching gears, so that's the main way to transmit these viral pathogens in a way that will decimate your colony to cause it to collapse. There are other ways that pathogens, not just viruses, but bacterial and fungal pathogens can be spread around a colony as well. The main ones being through a process known as trophallaxis. So that's basically when the bees are exchanging resources with one another. So you have a forager come into the hive and she's unloading her nectar stores into a bee that's job is then to store it in the colony, and that forager can then leave again. So it's kind of like a handing off of supplies, but with your mouth. That's what bees do. So that's obviously gonna have some issues, right? With kind of spreading disease and pathogens, especially with a lot of them focused primarily on being successful in the gut, where those resources are coming in and out of. So you can have a lot of these pathogens be inside the actual nectar or inside pollen grains. And so whenever the bees are exchanging resources, that's an easy way for them to transmit to one another. There are bee STDs, and they can be transmitted to queens from the sperm of a drone, and can be transmitted from the queen to her eggs. And I'm just going to kind of like go into that just a little bit more. So the transmission, general definition, transfer of a disease from one individual to another. So we have vertical, which I said before are the STDs, and it's from mom to baby. And then you have the more common one, horizontal, which I like to say is similar to when you're giving someone a handshake. It's from individual to individual. So adult to adult, or from adult to larvae. So for the queen, she can actually be infected with a virus, and that virus can actually be on the outside of each of the eggs she lays. And when the larvae hatches out of the egg, it will then be fed the brood food, and it can enter into the larvae that way. So you can have entire colonies become infected with something just because your queen is infected. So if you have the top kind of sprinkling down to the bottom, then no matter what you do to kind of remedy the worker population, if your queen's infected, it's going to be a continuous problem. Some of the viruses that can be transmitted this way from mom to egg, from queen to egg, are deformed wing virus, black queen cell virus, and some of those paralysis viruses I talked about. We do see a lot of symptomatic bees that are infected with DWD this way, so it is a common issue that we have to kind of think of our queen quality when thinking about how to deal with these pathogens. Like I said, bees get STDs. Who would have known? So you can have drones that are infected with, for instance, deformed wing virus, and they are just as competitive as their non-infected brothers, and they can infect queens that way as well. It can cause queen failure too, depending on the pathogen and how high the pathogen load is. And deformed wing virus and Israeli Q paralysis virus, that one that I said is associated with colony collapse disorder, are just two that can be transmitted from the drones to the queen. So for the direct transmission, like I said, trophallaxis is the #1 way that you're going to have pathogens spread around a colony. Other things to keep in mind of why we're going to have bees that are commonly infected with pathogens, not just because they're exchanging resources with one another, but also because it's a single queen that has thousands of individuals that are all genetically related to one another, right? So if you think of it in human terms, if you have your entire family crammed into one room and you're all living on top of each other, then if one of you gets sick, most likely everyone's going to get sick in that area, right? Especially when a confined living space such as a hive, and it just creates this perfect environment for disease transmission, especially in an overwintering period when the bees are confined in that space for the winter. So what I kind of like to do with beekeepers, especially because after a lot of meetings and stuff, I have people say, well, I think my hive's infected. I'm like, okay. They're like, what is it? I'm like, I need you to give me more. So what kind of the things we go through and trying to figure out and troubleshoot what's wrong with your colony, especially if it's something that is impacting your apiary and you're seeing it systematically. You kind of have to say, okay, what are the symptoms? Who is infected? Is it the brood? Is it the adults? Is it both? That's going to give you a really big hint about what you're dealing with. When did the symptoms appear? Time of year is a huge thing about when certain pathogens are more prevalent, and I'll go into that here in a bit. Also, where are your hives located? So, what part of the country are you in can also be an impact of what pathogen you're dealing with. From there, when you kind of have an answer to some of those or all those questions, then you can have a better guess of what you're dealing with. And once you identify the pathogen, you can say, okay, well, why do I think maybe this disease outbreak is occurring? Is it because there's nothing flowering right now and I'm not doing any supplemental feeding? Is it because I have a new neighbor who really doesn't take care of their colonies? What could happen? And then if you know the biology of the pathogen, then you can know how it's spread and how you can treat it. So like worst case scenario, knock on wood, I don't want to even wish this on any of you, but if American foul brood happened, then you want to have the notion of like, well, I can't use antibiotics, that's not going to be efficient, only if it's a European foul brood. So knowing what you're dealing with and the biology of the pathogen will help you with treating it. Speaking of treatment, typically if you have a strong, robust colony, high worker population, high food stores, a good quality queen, and they're not complete shade, then you typically can have colonies take care of themselves. They can usually clear out infection pretty well. However, in times of stress, especially during pollen dearth or when they're ramping up for overwintering, or if they've just swarmed and the worker population has been cut down, or if you are combining or splitting, then that might not be the case for them being able to take care of a disease outbreak themselves. So that's when you say, well, should I re-queen? Is this a queen issue? Or is there a treatment that I can apply to my colonies? And with all that, you can prevent future disease outbreaks and kind of help your colonies out. So I talked about who is infected is a big thing. A lot of these pathogens, not so much the viral, but a lot of the fungal and bacterial pathogens do target the larvae. So learning how to not just look at your adult bees, but looking at the larvae, looking at how they sit in the cell, looking to see what color they are, is there an odor that seems a little off. Paying attention to your larvae is going to be a big help in you catching things early. So for instance, this is what you all want to see, right? A very compact, solid brood frame. And if there is gaps missing, they're pretty uniform. That means they're all emerging at the same time, right? So the queen laid 2,000 eggs, 2,000, 2,000, 2,000. So even if you have a frame that looks like it may be missing a lot of space, as long as those spaces are pretty uniform together. So when you have this kind of shotgun pattern is kind of what we refer to it, where it just looks like, and I'm sorry, I know it's a little difficult to tell. So basically it just looks like you fired a shotgun and just the brood are just everywhere randomly and the larvae is randomly scattered about. There's all these empty cells and there's really no rhyme or reason to that. That typically means something's wrong with your brood and the bees are removing those larvae and taking them out and throwing them outside of the colony in a way to prevent future disease or further disease spread to the other larvae. So as they sense something's wrong with the larvae, they take it out, which kind of gives you that weird spotty pattern. So having spotty brood is indicative of one of these brood pathogens or with high varroa mite levels in your colony. So typically if you have your brood frame, you want there to be 2 times more larvae than eggs and 4 times more capped than eggs is kind of the ratio people go by. So yeah, so this, if your frames look like that, they're on their way out. You do no longer have a worker population that's going to replace the new ones. That colony is set for collapsing. So some of the ways that bees can take care of pathogens themselves. So they have evolved ways, even though they are very genetically similar and live on top of each other, there are ways that they can help prevent disease outbreak. One of it is grooming. And grooming is actually one of the biggest factors as far as removing varroa off of the adults. So they'll groom themselves and then they'll groom each other. And that helps if they have a high rate of grooming, then they're typically able to kind of like brush off the varroa off each other, which is kind of interesting that grooming behavior is genetically linked. Some bees are more cleanly or have higher cleanliness than others. So they can be bred to have more hygienic behavior and have more grooming behavior to help fight against varroa. And that's some of the research that's being done now, is to create those genetic lines. We also have bees whose entire job for a period of time are being undertakers. They're the ones who kick out the infected larvae. They're the ones who will drag an infected sister from the colony and throw her outside, which is kind of sad. But they're also the ones that if you squish a few bees and they fall in the hive, then they'll take those bodies and throw them out. It's a way to keep their living space clean and free of disease. Also, bees have what is known as self-sacrifice. They're altruistic, which means that they will sacrifice themselves for the greater good. They can detect to a certain level if they're sick with something, and they will actually remove themselves from the colony and will either become foragers earlier in life or just remove themselves entirely, even if they're nurses, and will die outside the colony for the sake of their sisters and family. So that's what's known as social immunity, taking care of the group. I mentioned hygienic behavior is the kind of focus right now in the research world of breeding for, where bees can be selected for realizing there's something wrong with the brood. That's specifically what the hygienic behavior is. The bees that are tending to the larvae will go over a cell and they'll notice and detect that something's wrong with that larvae and pull it out. That is not the same quality for each nurse as far as detecting when something's wrong. So it is a heritable trait, so you can have bees that are more likely to do that removal than others. So yeah, so I think, so for social immunity, this is found in eusocial organisms. So bees, ants, ones that have typically a queen and then a lot of related workers that all work together as one group. And they have different ways to combat pathogens, some of the ones I mentioned already. I'm not going to go into all of it. But some of the really cool ones include propolis, hygienic behavior, social fever is a really cool one. They use this not only to up the temperature of the colony to kind of kill off some pathogens that are not heat resistant, kind of similar to how we get a fever that can kind of like combat whatever we're sick with. Bees can have a social fever where they'll vibrate within the colony and raise the temperature of the colony as a whole in order to kill off certain pathogens. But they also do this as a way to attack a predator that was a big scare a little while ago, which is the Asian hornet. This is a behavior done by mostly Apis cerani, so the Asian honeybee, to fight off predators. The European honeybee usually only does it whenever they're inside their colony to raise the temperature of the colony. But the Asian honeybee has developed it in a way to kill the Asian hornet. So what they'll do is when they detect a hornet, they'll ball around her, similar to when they're balling a queen. They'll ball around the hornet and then they'll start to vibrate their wing muscles and they'll essentially cook the hornet alive. They have a 1 degree more tolerance, heat tolerance than the hornet, so they'll kill the hornet this way. Of course, a lot of them die to stinging from the hornet, but it's a very effective way to kill it. If you look up videos of it, it's really cool. This I wanted to throw up here because I just think it's very cool. So if you're unaware, what task a bee does in the colony is dependent on how old it is, and this isn't very strict. This has some plasticity. So they will be nurses earlier in life and foragers later in life as a rule. And then there's different roles that they fill in between. So undertaking is around the halfway point. Then comb building is the last nurse inside the colony role they have before they become workers. What's cool though is that bees have become infected with, for instance, deformed wing virus, Israeli Q paralysis virus, or nosema, will actually become foragers much earlier in life than non-infected individuals. So it is not known if this is the pathogen impacting the host or if it's the bee itself removing itself from the hive in order to not infect your sisters. We don't know which one it is yet, but it's seen very commonly where infected bees will become foragers earlier. The problem with that is that you have now a much larger forager population, less nurses, and you can kind of have an off-balanced colony dynamic. And if you keep having infected individuals become foragers, then your colony can collapse as a result because you don't have enough nurses tending the new population. Sorry, I'm going to take a drink of water. Any questions so far?
Alex, let me ask one question.
Awesome. I was like, I can't be doing that great a job.
Do these get caused just by the environment outside of the bees, or is there something the beekeeper does that causes some of these?
These, how pathogens are spread?
Does something the beekeeper do cause a pathogen to be there or to spread? Or is it just something from the environment that then has to be dealt with?
It's both, and Chris, I'm going to answer your question here in a bit. I'm sorry, I'm going to give you the just wait for a second answer, but the answer is both in a short sense. So one of the social immunity aspects I talked about was propolis. You guys are all familiar with propolis. I'm sure it's sticky stuff we're battling all the time. And I know I've personally been so annoyed at trying to open up a colony that I'll be scraping off the propolis and I'll fling it away because it's just so annoying to try and unstick some of those boxes. Not the best practice. Propolis actually, not only does it air seal a colony and help keep out environmental factors like cold wind or other pests that can crawl into spaces. Not only is it a good architectural resin that the bees use, but also it's been found to have antimicrobial properties. There was a study where this graph, what I just want you to get from it is that in red, they treated workers with propolis that were infected with nosema, and then the blue was ones that weren't treated. And what they found was that the bees that were treated with propolis, so they were fed the propolis and they had it in their environment, they had a higher immune response to being infected. So basically they were better able to combat the pathogen, if that makes sense. So bees that have propolis in their diet and in their environment are better able to have a robust immune response and better deal with pathogens. So keeping propolis in your colonies, even if you scrape it off, I always just put it back onto the entrance or nearby so that they can collect it again. It is a time consuming and it does take a lot of energy for them to create that propolis, and it's kind of a win-win for them. So keeping it there is a good thing. How you arrange your colonies is also very important. So this is kind of me now getting into the what can we do as beekeepers to combat a lot of these pathogens. I know, especially with viruses, it's like, sorry, there's no cure, deal with it. But there are ways that you can kind of help your colonies be better equipped to deal with it. So for instance, I'm going to use my mouse here. This was the apiary setup for this experiment. They had one apiary where they had all of their colonies on a single hive stand in a row. Pretty typical for what we see in our yards or see in commercial or sideliner beekeeper operations. But then they had another array where they basically put all of the bees in a circle, and I believe it was the diameter was about 100 meters. So they had all the bees in a circle with the entrances facing outwards. And what they found was that bees that were in that array had a lot less drift and a lot less pathogen transfer was happening. So they basically set out a bait station and they kind of tracked this colored dye and saw how much that dye was transferred between different colonies. And then they also set up an infection experiment where they were seeing, well, if we infect one colony, how likely is it to spread to the others? Well, when you have all of your colonies basically one on top, like one next to the other, those entrances aren't too far apart. The room for error for a bee that's become a forager early in life because it's infected with something to drift into a new colony and then infect that new colony whenever it does the prophylaxis with a new worker is much higher. We know from studies that bees that are carrying pollen or nectar or whatnot, even if they're not originally from that colony, the guards and the other workers will accept that bee and they're like, well, if you're bringing food, sure, you can come in. So even if you have a bee from another colony drifting in and it's infected, if it's carrying a resource, then the bees are more likely to accept it and you have pathogens spread that way. Whereas if you have this array where the hives all have the entrances staggered, facing away from one another, you have way less drift, way less transfer of pathogens that way. I know it's not feasible for everybody, especially if you have a small yard. But just even staggering your entrances to face away from each other like that can help. So that's one thing we can do as beekeepers to prevent transfer. Another thing is the role of diets and making sure that our colonies are properly fed. So nectar honey provides the carbs for bees. It also can provide secondary plant metabolites and it's necessary for their energy. Pollen, it's their protein source, essential amino acids. I'm sorry I'm going over this fast because I'm sure you guys all know this already and I don't want to bore you. They need both, and they don't just need both from a single plant species. They need a wide variety of different plant species to meet their needs. So lots of diversity is important for honeybee health. So related to that, myself and another graduate student in the lab, we did an experiment where we used his expertise in nutrition and mine in pathogens. We were basically looking at, well, if we have bees infected with deformed wing virus or nosema, the fungal pathogen, we feed them different diets with varying protein to fat ratios. Are they better able to fight off disease? So it did involve me injecting a lot of bees. So that's sad, but it's science, I guess. What I just want you to get from this is our data. I'm excited about it, so I'm throwing in my own things. But this is our data, which we basically had our 4 different diets, which bees that got no diet whatsoever, a high fat diet, a high protein diet, so 40 parts protein, 10 parts lipid, and then a medium diet, which is at 30-20. And what we found were that bees that were given a unbalanced diet, so they had a lot of fat in their diet or even a lot of protein, which is odd because a lot of our supplements that we buy are known for having very, very high protein ratios. If you have one of these more unbalanced diets, bees actually had quite a bit of mortality when infected with DWB. So for instance, bees fed that high protein diet of 14-10 right here. You can see in red that by the end of the experiment, which was only 2 weeks, we had about 30% of our caged bees die off by the end of the experiment, whereas it was up to 90% for our negative controls. Yes?
What do you mean by fat? What did you feed them fat?
So it was a lipid source. We used wasn't canola oil. It was linseed oil. We used linseed oil as our fat source. So we did create the diets in lab. It was artificial diets. That way we could control completely what was in there. Pollen, using pollen is not really an option because of how variable it is. But yeah, we used linseed oil as our fat source. And then we used forgetting now, a protein powder. And I can't remember off the top of my head what the source was. That's Pierre's thing, the nutrition part. I'm just infecting bees and counting how effective they are. But yeah, so basically what we found was bees fed a more balanced diet, which makes sense, right? For any organism, having a balanced diet of the right nutrients and the right macronutrients is going to be better off for bees, especially when they're dealing with these pathogens. So I thought that was pretty cool. And the takeaway is just making sure your bees are fed, especially during times of pollen dearth or during times when nothing's flowering and they're starving, then giving them sugar water or honey drinks is always a good idea. How am I doing on time, guys? Okay, I'm almost done, I promise. So here, I kind of wanted to throw this up. I think this was a special slide made just today, Chris. So this is kind of showing what some of the more prevalent pathogens are, depending on the time of year. So right now we're gearing up for the overwintering period, and more so a little bit earlier, but still now because we live in Texas. You're going to see high varroa mite levels in the fall prior to overwintering, and then associated viruses such as deformed wing virus are known to spike in the fall, and those are 2 of the biggest contributors to colony overwintering collapse. So making sure before you go into the wintering period that you have low varroa loads will also result in low viral loads and have a better shot of having your colony survive. So spring is a really big one. Spring season, once you emerge from the overwintering period, that's when you kind of got to look for a lot of the fungal and bacterial pathogens. That being because the colony has been stuck inside for such a long period of time. A lot of these gut pathogens where normally they could go outside to defecate, get rid of the pathogen that way. If they are defecating inside of the colony and then stuck inside for the overwintering period, it causes stuff to spread much quicker. Also, just because of the rainy season in the spring, you can have a lot more fungal and pathogen bacterial growth that way. All right, so going into the overwintering period, you should make sure your colonies have sufficient food stores, use hive reducers to limit robbing, and then also monitor and control varroa loads, like I said. So bees will be able to detect when it's time to overwinter based off of the shorter and colder day periods, and that causes a reduced amount of foraging, which increases the amount of the forager pheromone inside of the colony, and then that causes the bees to be like, oh, it's winter. Another way is reduced floral resources means reduced nutrition. If they have less pollen, they're going to rear less brood. There's less brood pheromone, and the bees are like, oh, it's winter. So they're going to stay inside their colony until environmental factors, temperature, causes them to emerge in the spring. You just have to make sure that they have, I would say the biggest one is varroa and then food stores. The reducing robbing just helps with like not having infected bees spread between colonies. All right, so the big takeaway summary for some of the best beekeeping practices to limit the transmission of pathogens, stuff that we can control somewhat, is where you place your colonies. So like I said, having them with the entrances facing away from each other from ones that are right next to them can have a very big impact. Also, having your colonies in more sunny areas can also help against a lot of pests, like small hive beetles, for instance. If you have colonies inside of very shady spots, that does encourage small hive beetle populations. So where you put them is important. If you do come across frames that have something wrong with them, they have foul brood, they have nosema, whatnot, don't just throw them off to the side and then reuse them later. A lot of these things can persist for a long period of time. So the best way to get rid of bacterial and fungal pathogens on your stuff, if you do want to reuse your beekeeping equipment, is to throw it in a freezer for, always safe to be like 2 weeks. That typically kills stuff really well. And then you can kind of like scrape off some of the bad stuff and then reuse it, or even just reuse the cells and the bees will clean it out. But by then, all of the pathogens will be dead and they won't be able to replicate. Not the same for viruses. Those will persist even if you put them in -80. So. Strong colonies are better able to deal with stressors than weak ones. So like I mentioned in the past, stronger colonies can sometimes deal with an infection on their own. Strong equals high population sizes, good queen, and good amount of food stores. If you have those three, they can pretty much clear up a lot of the stuff on their own. If they don't have those three, then maybe you need to look into, especially before the winter period, combining your weaker ones into stronger ones, putting frames of honey maybe have stored in the freezer or something into weaker colonies, etc. Yep, I just said that. Making sure you feed your bees, do keep an eye on them. I mean, sugar water is not the best thing, but food is better than no food. McDonald's is better than starving. So just putting sugar water in there, especially during times when we have weird seasons where we have a drought and nothing's flowering, for instance. And check and address varroa issues. I know I never try and get into recommending certain products. In fact, I will not. I'm not allowed to anyway. But however you deal with varroa, just make sure you're checking your varroa levels every now and then, especially this time of the year. This is a very important time to do so. Maybe more so earlier in the season so you can treat it, but addressing your varroa issues is super critical. And then re-cleaning is always an option. If nothing else seems to work, your queen quality might be what's kind of impacting your hive. I don't think I really have time to, I just threw this up here because some people ask me like, well, what can I do against my viruses? We'll go through it real quick. Who wants to answer this for me? Are there antibiotics that can be used to treat my colony when infected with viruses? Can anyone think of one?
Antibiotics don't cure viruses.
Thank you. Love that answer. Thank you so much. You'd be surprised. I gave a similar talk to an undergrad class at Texas A&M, and they were throwing all sorts of stuff at me, and I was a little bit sad. So thank you guys. All right, so currently there is no commercial option for viruses. There is research looking into priming a bee's immune system through the queen. Not going to go into that right now, but if you're interested, I can talk about it later if you want to come up and ask. So the question is, how do I deal with viruses then in my hive? You deal with varroa, always the issue. Viruses really weren't a huge issue for beekeeping until we had the introduction of varroa, and then they became that dirty needle that just set everything off. So deal with your varroa and you'll basically be able to deal with your viral population sizes. And with that, I hope I didn't go through too fast, but I'll take any questions. Yes, Anissa.
Alex, can you talk a little bit about queen quality overall?
Yes. Okay, so queen quality overall was the question. Can I talk about it? So queen quality, how we would measure it in previous studies that we've done in the lab. Liz Walsh, if any of you are familiar with her, she would look at retinue size. So workers can actually detect how good quality a queen is and be more likely to tend to her. The reason being is queens emit what is known as queen pheromone. It's a pheromone specific to just her, and it will waft through the colony, and it triggers a lot of different events such as needing to swarm or needing to build out more comb. So how much pheromone she emits does impact her quality. So one way you can look at that is how many attendees does she have? Is she surrounded? That's typically the sign of a really good queen. If it looks like she's solo and no one wants to deal with her, that's literally the case. No one wants her. So that's a big one. If she's, how much egg laying she's doing is a big one too. So queens that are sick or have something wrong with them typically will not lay as often in a minute time period. So just watching how often she's like putting her abdomen in a cell will give you an indication as well.
So how many eggs will a good queen normally lay in a minute?
It's 2,000 a day. I can't do the math right now. But whatever that conversion is. But yeah, so size doesn't really give you too much of an indicator. Genetic stock is a huge one though. So where you get your queens, what stock they are, can impact quality as well. Yes.
You mentioned putting your frames in the freezer for 2 weeks.
Yes.
That's for the bacteria.
I used it mainly for nosema. I had a nosema infection, that was the time period it took for the spores to no longer be able to replicate. They basically they were present, but they weren't going to get me sick. That number is, don't quote me on that, but that's just what's worked for me. And yes.
So you were talking about hive placement in the circular, which I love that idea, but I've always been, told to face the hive entrance to the south. So how do you do that? You just do 2 half circles and face them on the south, or it doesn't matter?
No, see, I've gotten that question before too. And can you explain to, I guess, everyone, why were you told to have your hives facing the south?
To protect them from the winter.
But we're
Yeah.
we're in Texas, and
Okay, then, so can I move my existing hives?
yes, you can, but what I would suggest with that, there is kind of an art to moving a colony that's already established. Do it either early morning before your foragers leave, or seal them up at night when everyone's back in. That way you don't, when you move it during the day, lose that forager population. So if you do want to move them, that's way. I know hive stands are super easy, but putting them on cinder blocks is just as well. You can make fancy tables if you're fancy, but yeah, so facing them southbound, a lot of people say that too because of like winds and stuff, but it's not too much of a deal here. Yes.
Do queens lay less
I don't think I can't confidently answer that. I'm not sure.
Can you repeat the question?
It wasn't
Can you repeat that question?
Oh, I'm sorry. The question was, do queens lay less often in the evening, right? Yes. And I said, I'm not sure. Typically, I don't try to stick around that long. But I was involved in a project where I had to count the number of times she was laying. And let me tell you, like, I got through a few hours, I'm like, okay, I'm gone. So, yes. Yes.
So at this time of year, I've been reading and trying to understand how to balance out hives. So I have 2 weak hives
Mhm.
and 4 strong hives.
Okay.
So shall I combine, but isn't there a danger in combining a weak hive, adding it back to a,
To a strong one?
yeah, because of pathogens and everything else?
Right.
Why is it weak? You know, the
Exactly.
reason it's weak, do you want to keep it, or do you just let it go into the eons? And
Oh, do Darwinian beekeeping mean?
yeah. So combining hives, weak with strong, isn't that?
So for, I guess, the people on Zoom, the question is, should I be combining weak colonies with strong colonies, especially if I don't know what made them weak in the first place, correct? Okay, so yeah, knowing what made them weak is a big factor. If it's just, we've had our apiary manager say, Gene, you guys know PT, he's now in California, but he would say, just put them together, they'll figure it out. It depends who you talk to. If you're just combining frames, as long as there's not an issue with brood, if it's just food frames that you're combining maybe from the strong into the weak, that's typically fine. If you're trying to just combine the entire colony together, I would look to see what the issue was. If you can, I would look to see what the varroa loads are in that weak colony first, if you can, and then just kind of look at the brood and see if there's anything wrong. If there doesn't appear to be issues with brood, and if the varroa population is low, I'd say you're pretty safe to combine, if that makes sense.
Yes, except it's hard to check the rural population when it's really a minimal colony at this point. We know it's on its way out.
Yes,
Right.
yeah.
So
Then at that point, I would still combine them if it's that small of a population. I don't know, people with more beekeeping.
You can't find a queen and you see just a little bit of brood left. Just take all their resources and give.
I personally would, but people with more beekeeping experience. You guys want to chime in?
I do that too.
A stronger colony is going to be better than a weaker colony. So when you combine that weaker group there, they're going to become part of a stronger colony. And whatever condition they have will probably be helped by the even stronger colony.
Okay, I agree. So yeah, I would go ahead, combine, and you're going to have a better chance of all those bees surviving if they're stronger anyway.
Do you do that newspaper thing where you put the newspaper between?
I don't. I just smoke the crap out of them. And they're usually
Is that a
fine.
scientific method?
No. They're fine. Yeah.
Screen potted boards or solid? What's your preference?
I'm sorry, can you repeat it?
Screen potted board versus solid. What's your
Okay, the question is screened or solid bottom boards. We use screen, but then a lot of the times during the wintering period, we'll put one of those light counter, perforated like whiteboards, you know, we'll slide those in there to kind of just help with maintaining the temperature within the colony, but we typically do screen. Yes.
So I'm new to all this. I'm learning.
I know, I loved the note taking. I was
But,
very appreciative of that.
Yeah. Concern over fire ants because they like to get into everything.
Don't get me started. I love, okay, so you just hit on one of my passion projects, which was my first ever chapter of my dissertation was looking at the interactions of ants and honeybees. And I did work with some of the beekeepers here in the area about that. But yeah, one of the big issues was we saw fire ants a lot of times in colonies. And my question was, how are they interacting with the bees? And then are they picking up viruses? So I did detect viruses, mainly deformed wing virus, in primarily fire ants, because they'll go into those colonies, they'll rob out the larvae and they'll also eat the food stores and they'll pick it up that way, and then they can mechanically transfer to other colonies that way. How to deal with fire ants? There's a multitude of ways. It just kind of depends on what you want to use personally. Even just putting hive stand legs in soapy water can be an efficient way to do it. You just have to be careful about rain, and it does dry out over time. Some people swear by cinnamon. I've never used that myself. But yeah, just making sure that if there is a fire ant colony nearby, you can just spot treat that and it'll be fine.
Amdro is my favorite go-to.
Yeah.
So as long as it doesn't hurt the bees.
No, you can treat the fire ant colonies and it won't impact the bees.
My neighbor claimed that when he put cinnamon out, the bees were gone. All 3 hives.
All 3 hives. Did he put it in the
I
colony?
have no idea.
Okay.
He said it was somewhere around. And I was using a toxic product away from my,
I've heard lots, like cinnamon is one I've heard commonly. Some people use essential oils. I'm not gonna put any backing on that. What I literally just do is, we just put the legs in soap water. But just getting your hives off the ground will already make a huge difference. No, I love it. That's not what I thought you meant when you said you're right.
Can you talk to us about the cohorts and the wrapping and all that? Because we did have that.
Right, okay, so the question is to talk about wrapping the colonies for winter, right? I personally have no experience with that. I've only done beekeeping here in Texas, and we did not wrap our research colonies for 3s. Honestly, a lot of the U.S. Department of Agriculture has some really good resources, especially the ones that are farther up north. They do a lot of research looking at not just only wrapping, but like creating those big buildings to put your colonies inside of for regular temperature. Sorry, I personally can't give you any information on that. I'm not too knowledgeable about wrapping colonies. I don't know if somebody else would like to chime in, if anyone has. Chris, where's Chris?
We don't wrap colonies in Texas.
We don't wrap colonies in Texas.
Winter colony.
Yeah.
Now, last year, you may have wanted to last February.
Yeah, that was a weird freak incident. Did I answer? I mean, that wasn't an answer, but sorry. Anything else?
Thank you.
Yeah, no problem. Did we have chat questions? And if you have other ones, feel free to just come find me.
Alex, we want to thank you for coming and speaking to us tonight.
Yeah.
So I just, it's always fascinating to me that somebody so young is very interested in the sciences, and
Oh, yeah.
especially a female, so we wish you luck. Now, before you go, last month, at our board meeting, and then we discussed this at our meeting, we decided as our board to donate $1,500 to the
Nevin Weaver, Honeybee Excellence.
we donated 1,000. I thought I was talking loud. We donated 1,500 to the Devin Weaver Honeybee Excellence Fund. That money goes to support Dr. Rangel at Texas A&M University. It helps to support her, what is it?
Well, let me tell you, first of all, the money isn't used, the interest on the money is used so that the capital stays there forever. And the second thing is that our aim at Texas Honeybee Education Association was to raise 100,000, and we're within a few dollars of that. So we've done some excellent work for
Thank you, guys.
A&M Lab.
So part in
And
part
I
of
told
that.
Dr. Rangel that that meant we wanted people like Alex here to talk to us.
Yeah.
So this
Not
is
bought
this is your product of Dr. Rangel's teaching, so we're very appreciative of you coming. So before she goes, Dr. Rangel was kind enough to send a thank you note, which to even get a thank you note in the mail just means a lot. But I want to read it to you guys because for us, our group is about learning and education. And so I just want to say, I just want to read the thank you letter. On behalf of Texas A&M University Honeybee Research Program, I want to thank you for your donation to the Nevin Beaver Excellence Fund. Your contribution will help us reach our goal of funding graduate research for years to come. Thank you very much. So I just wanted to share that with you guys because to just even take the time, Jimmy even got he got a thank you note too. So the fact that she took the time to write these thank you notes and the fact that we're also donating, it means a lot. So thank you very much.
Of course, thank you guys for your donations. It supports not only myself, but the younger students that are coming in and have a lot of passion and ideas. So thank you for that.
And last month we talked about the license plate. So the Texas Honeybee Education Association, they have a lot of scholarship programs. So for every license plate that is purchased through the state of Texas, they get $22 towards the scholarship fund. So before our meeting, or before we started the speaker, we talked about the TBA meeting. So our group always sponsors a gift for the live auction or the silent auction. And so we as an organization spend around $100. And so what we're doing this year is we're donating a set of steak knives. These have a honeybee on the handle. And so we're donating these and the proceeds will go towards the TBA and help with education for beekeepers. And then we also, every meeting, for those of you that haven't been here to the meeting, we do a door prize. So, and I'm gonna ask for you to pull one ticket. So what's the number?
It's with the last two numbers. 462.
So if your ticket is 462, we do have a door prize. So thank you very much. So the door prizes, if you have, like at our prior meetings when we had a lot more people, if you have eggs or you have honey-friendly plants, something like that, we use donations for door prizes. So if you do have anything that you would like to share with other members to be used as a door prize, you can bring that to our next meeting. So thank you very much. And we want to, does anybody have any other questions or? Okay. We want to thank everybody for coming and thank you very much. Everybody have a great weekend. Oh, wait. You know, as I get older, I have short-term memory. That's at all either.
How do you think we feel about that?
It's been a long day. The other thing, Jimmy's wife was kind enough to make a centerpiece for the TBA meeting. It's the dinner that they'll be having. Okay, so every club donates something. This Jimmy's wife made, and our club will be donating that for one of the centerpieces. We also, Linda, who does our programs, was kind enough to donate this.
This beautiful picture.
And that will also be used in the live auction or the silent auction. So if you're interested in going, those will be available for the silent auction. So we want to, did I miss anything? Did I miss it? Okay. We want to thank everybody for coming and you guys have a great weekend.
Thanks for your question. I got so excited. I was like, that's my research. But I hope I have no idea the answer too well. The answer was